Welcome. This is week 6, lecture 4. So we'll continue with the demonstration of using command line interface in this lecture as well, but we'll focus on the short read archive and the toolkit that is associated with it. So it's also known as the SRA toolkit. Right? So before we go into the SRA toolkit, a little bit background about short read archive or SRA. So it's again a part of your NCBI database. But it's a very specific part of it where all the short read data and now even the long read data is being stored. So you can think of it more like an archive for all the different type of next generation sequencing, whether it is Illumina, whether it is PacBio, whether it is Nanopore, all of that data is stored here. So similar to the other NCBI dataset, there are a lot of good resources to help you get started, to know more about the database. There are also a lot of tools and software to access the database, to download things from here, and there are various other related resources where the metadata associated with these sequencing datasets is stored. So for example, the bio project and biosample, these contain the metadata, and also there is a submission portal where you can submit new short read data that you generate, that can be submitted in the submission portal. Right. So let's look at one example. Let's say you search for the word chicken in the short read archive. So what it will do, it will list out all the next gen sequencing datasets associated with chicken. So you see that there are so many datasets, so around 2 million datasets associated with just chicken. Then here you have a tree where it specifies what is the species from which these data are derived. So only 64,000 of them are actually from Gallus gallus. But then there are some which are chicken gut metagenome, there are some which are actually bacterial genomes which have been, or bacterial sequences that have been derived, maybe with some source being a chicken or something like that. So that's why when you search for chicken, so you get some of these other species as well, like Salmonella or Campylobacter, and few other tags are also getting picked up. But just with respect to Gallus gallus, there are still 64,000 datasets. But then when you see where are all these datasets distributed, you see that most of it is not most, but quite a large number is associated with Gallus. Then you can of course subset it. So for example, here we have DNA, RNA, or you could have genomic or exomic. You can have paired end or single end data. You can also have different type of sequencing platforms. You can subset it so that you only focus on this Gallus gallus dataset. So you click on it, then you get to a subset of that data where only these 64,000 records are shown. And here what is the search string? The search string has changed a little bit. So it's added this AND operator, and it's also specifying the species name with the taxonomy ID for this, so that only those datasets associated with chicken Gallus gallus is shown here. So even that is 64,000 records. So in that, if you see, there are some epigenomics datasets, some RNA-seq, some genome. Then there are the file type is again very diverse. There are some files stored as BAM, some are stored as fastq, some are stored as Illumina native, that is before it is converted to the fastq format. There are some stored in that format. There is also some 454 data stored in SFF format or SRF format. There are also some aligned data, maybe in the form of BAM or CRAM. But again, all of this data is not from one platform or just short read data, but it includes some long read data as well. So all of these 64,000 records or 64,000 datasets, you can see where they are coming from. Then again, of course, if you want to focus on a specific data in this, only not look at everything, but look at specific data, you can again choose which filters you want to add. Maybe you want to look at only RNA, or maybe only want to look at DNA. You can click on it and that filter will get applied, and only that part of that subset of the data is going to get shown. Right. So we talked about data organization in SRA earlier as well. So we said, okay, there is sequence read archive, and in sequence read archive all the data is stored in the form of experiments. So in each experiment you can have multiple runs. So you can have each run could be a pair of fastq files or a BAM file. So if within an experiment you can have multiple runs. So they could be replicates of the same experiment, or they could be the same data run on multiple lanes, so that to get a higher coverage. So these runs are all associated with a single experiment, and the experiment has specific biological information associated with it in the form of biosample. So the biosample is stored on a different database, where description of the sample, what is the taxonomic details of this sample, and other metadata associated with the sample that is being sequenced, or the experiment that is being sequenced, is stored in the biosample. And there is also a higher level hierarchy, that is your bioproject. A bioproject can contain multiple such biosamples, and in your bioproject you describe the overall goal of your project. Right? So what are you trying to achieve by doing all these different experiments? What are the grants which have funded this particular project, which have allowed your data set to be generated? What are the publications that have come out of this particular project? All of that is stored in the bioproject. So if somebody wants to use this data set, looking at the bioproject can be quite useful, because then okay, these are the previous work that has been done, and that's what has already been found out from these data sets. Then you want to reuse this data set, you probably want to look at this and see, okay, these are the papers which are the source of the original data. So this gives you some idea of data organization in SRA, whether it is a genomic data, transcriptomic data, or some type of epigenomic data, whatever be the assay, the data, the fastq data, the raw sequencing data is stored in the short read archive. So if we look at one example of a bioproject, we can look at an example dataset which we generated actually just last year. So in this we actually sequenced the genome of a chicken breed native to India. This is called Kadaknath. So this is the project where this is stored. Then there is a publication associated with it. So details of the publication are mentioned. Also it mentions when this project was registered and who is the owner of this project, or who is the owner of all this data. So you see that it lists out the name of Isa Bhopal here. Then if you look further, it has more details, right? Like it tells you how many experiments are there. So you see that there are 21 experiments. So it's not, one project is not just one experiment, it can have multiple experiments, and within that you have 12 samples, right? So although there are 21 experiments, so there are around 12 samples. So some of the samples have more than one experiment associated with it, and it also tells you the total amount of data that is generated. There is a 513 gigabases, or if you want to look at it in terms of TBs, terabytes, it is around 0.17. So this tells you the overall data set that is there. So this is given in the bioproject. So you can search for whatever bioproject you want on the bioproject database within again NCBI. But then if you want to look at these experiments, so you can just click on this, then it will show you what are all the experiments that are associated with it, or you can click on the biosamples, it will show you what are all the biosamples associated with it. So here you see there are 12 biosamples. So it mentions what it is. It's saying that this species is Gallus gallus, but then also it gives you a biosample ID, then it tells you what is the sample experiments associated with it. Then it also gives you some kind of a description about the sample, what type of chicken breed is being sequenced, and within that what subtype is being sequenced, right? So these are morphs within the breed. So these morphs are again given some name. So these details are mentioned when you go into the biosample. But when you go into the biosample in greater detail, it will have more details like where it was sampled. For example, this sample was sampled from Jabua district, or you can also get the coordinates of the location where it was sampled. Then it tells you whether it was a male or female, which tissue was sampled. In this case, liver tissue was sampled. It also mentions the details of when the data was first released to the public, were there any updates to it, and what time it was released, right? All of these details are provided. In some cases you may have additional details as well. So when we say sequencing data set, all this metadata which is linked to it in the biosample is very useful, because without this metadata you don't know where this sample was obtained, you don't have enough context to make sense of this data. So having this kind of attributes or having this kind of metadata is extremely useful, and SRA provides a very nice interface where such metadata is very clearly linked to each of these sequencing data sets. So if we go to downloading these data with SRA Toolkit, so we will use this SRA Toolkit. So first we have to install the SRA Toolkit. how do we install it? We can just download it using wget. We earlier used something called curl to download the NCBI datasets command. So here we are going to download the SRA toolkit. Again, it's available as a binary in an archive. So you can just use wget to download it, and you can specify where it has to be saved. So you save it in an archive called SRA toolkit.tar.gz, then it will download it. Then you can just extract this archive using this command. So this is first just the download. Then once the download is done, you get the binaries. Then you want to extract it. So you use tar with this flag minus vxzf, and then you give the archive name. So then the archive will be extracted, and all the binaries which are there within it will be extracted into this folder. So you see that there are various folders. So there is a folder for SRA, there is a folder for NCBI, there is another folder for binaries. So then all the binaries are extracted. So once the binaries are downloaded and once they are on your system, you can go ahead and use them. So this is one way to install, similar to curl, where a binary is available, you just download it and you start using it right away. Right, so coming back to the data that we discussed about. So this is some data that we generated. As I mentioned, this is associated with this paper where we looked at various aspects of chicken population genetics and things like that. So this is the experiment details which is there on the short read archive. What are the experiment details? It tells you who is the submitter or who is the data owner. It is providing that. It's also providing a description of the study, that genome sequencing is being done for this breed of chicken. It also is specifying what type of morph it is, what was the sequencing instrument that was used. So it was a Illumina NovaSeq 6000. So this is a type of Illumina sequencer. So this was used, and it was whole genome sequencing. So it's not just part of the genome that was sequenced, the entirety of the genome was sequenced, and it's a paired end type of library. So that is, it's not single end, you're sequencing from both ends, right? So you have a paired end library. Then this is the run number. Then this is telling you the number of bases that have been sequenced. So it's around 36 gigabases, and the size in GB is in gigabytes, and it says that it was published in 2023, right? So it gives you the details about this particular run of this data. So there are of course multiple runs of data associated with this project. So once you identify, okay, this is the run and this is the data set that is there, it's possible to download this data set and use this data set, right? So let's say we want to download the fastq file. So once we have downloaded the SRA toolkit, if you go to the binary folder, so where all the binaries are saved, there is a command called fastq-dump. So what this fastq-dump does is, if you give an accession number of a particular run, it will use that to extract the fastq file from SRA just based on the accession number and save it to your computer, right? So here we are going to do a test run. So we are going to just download 2 reads, not the entirety of the file, from this particular run, right? The Kadaknath genome sequencing run that we were looking at in the previous slide. We will just download that and we'll print it to the screen. So when we do that, it prints out these 2 reads to the screen. So it's in your standard fastq format. So if you remember your fastq format from one of the earlier lectures, it has 4 lines. The first line is the header line. So the header line is starting with this @ symbol, and then in the second line you have the sequence. So the second line is spread over 2 lines because the length is quite long. It's 300 base per read. Then there is a third line which is again giving you the header information, and then there is a fourth line which gives you the quality score. So this is the sequence. So this is the quality score, and this is your header. So in the header you have details about the sequencing machine that was used, what is the length of the read, what is the barcode that was used, things like that, right? So using this kind of a fastq-dump command, we can download data sets from SRA. So if you don't want just the 2 reads, you don't specify this -x2 flag, and you don't specify the standard out, then it will just download it to your computer. So given any accession number, you can download datasets from SRA. So this is a fairly straightforward tool which allows you to download large datasets from SRA. Of course, you can put it in a for loop and things like that to download multiple datasets. So similar to how we looked at downloading multiple genes in NCBI dataset, you can also download multiple accession numbers using the fastq-dump. Right, so once you have downloaded your reads, the next thing is you probably want to map it to a reference genome and then see how this particular breed of chicken is different from standard reference that is there for chicken, right. So to do that, the first thing to do is you have to map the reads to a reference, right. So when we say map the reads, what we are doing is we are taking these individual sequencing reads, we are aligning it to a reference genome that is already available for chicken, and based on that we are generating your BAM files or alignment files, right. So to do this kind of read mapping, you need a read mapper. So we are going to see how you can use the BWA read mapper to do this kind of read mapping, and specifically we are going to look at an option called MEM, which is an algorithm which is within BWA. It's a type of read mapper. We are going to use that to do the read mapping. But before we start using the BWA read mapper, we need to install BWA read mapper. So unlike the previous examples where we just downloaded the binary, for installing BWA, what we are going to do is we are going to compile it from the source code. So we are not just going to download the pre-compiled binary, which is quite good if it it's possible, but it's also the situation that in many programs pre-compiled binary is not available, or pre-compiled binary is not compatible with your version of the Linux OS, right. So then you will have to compile from the source code. So it's always good to learn how to do this. So in the case of BWA, the source code is hosted on GitHub. So there is a link to a GitHub page. So you just do git clone. So then what it will do is it will just clone this GitHub website, right. This particular repository is cloned to your system. So then it will download all these files and folders which are there in this online GitHub. So once you have cloned it to your system, you change your directory to the folder where this repository has been downloaded. So once you go into that folder, you just have to type one command, that is the make command. So what make command is doing is it's taking all this source code and it is compiling them into binaries. So it will run all these different steps. So you see a GCC compiler is being called and it is compiling these different source code files. So these are the different C program files that have been downloaded from the GitHub repository. So all of them are being compiled, and you may get some warnings, things like that. Sometimes you may even get errors which you may have to fix, but then once it completes without any errors, it will give you the binaries. So then you will have a binary called BWA on which you can do all your, it will be your just like a binary which you download from the website, but here you have compiled it from the source code. So once you have this binary, you can go ahead and use this read mapper to map the reads. But of course remember that this GitHub link will be different for different software. So this is the link for BWA, and of course there is a help page associated with this on the web, so you can use that. And then once you download it, in most cases you'll have to go into that folder or one of the subfolders. So you may have to go into some folder called BWA or SRC in some cases, or in some cases you may have to compile within the BWA, then it will compile. Okay, once you have compiled, once you have your binary, you can just list the options that are there in your BWA command. We talked about this in one of the earlier lectures. You may have to use the -h flag or -- help flag, or in some cases you just type the program's name and it will list out all the options. In the case of BWA, if you just type /BWA, it will list out all the options that are there, right. So you see that there are options to index a genome, there are different options to map reads, and there are some options which are well suited for mapping short reads, there are some options which are better for mapping longer reads, right. So BWA has a bunch of different options for doing this kind of read mapping, and it's one of the more popular read mapping tools that's been there around for quite some time, and also fits well with many standard variant calling pipelines, and you can also think of it as part of some good practices pipelines. So BWA is fairly well regarded when it comes to read mapping. Of course there are various other read mappers depending on your preference and what application you're trying to use it for. You could use other read mappers, but the overall operation or overall principle is going to remain more or less the same. So the first thing that you're going to do with any read mapper is you're going to build an index of the genome, right, the reference genome to which you're going to map. You're going to build an index. So what we mean by an index, you can think of it something like this. If you have a genome, it's a very large sequence space. So if you want to map reads, you will have to go through that entire FASTA file and find where your read maps, right. So this can be very time consuming. So to make this process simpler, right, so that it is computationally faster and computationally less expensive, what you do is you convert the text file into a binary index. So when we say index, what you're doing is you're building a dictionary of sorts of where different k-mers are occurring. K-mers can be thought of as stretches of your nucleotide sequence of certain length, right. So then this is achieved through this command called bwa index, and then you specify the FASTA file, chicken.fa or gallus.fa, some kind of a genome FASTA file which has all the different chromosomes of the chicken genome, that is specified. So when you do that, what your indexing command is doing is, this is the genome, it looks for stretches of sequence which are the same, right. So this part of the genome and this part of the genome may have the same k-mer. So then it identifies all such k-mers. So for example, let's say the k-mer was T 6 times in a sequence. So then it's occurring at position 24, then it's occurring at position 139, it's occurring at position 1004, so on. So what it will do is it will build a dictionary saying that this particular k-mer is occurring at these positions. Then there is second k-mer which is this, so it's occurring at another set of positions. This is the third k-mer which is occurring at another set of positions. So it will build a dictionary of all these sequences and the positions at which these sequences occur. So once you build this kind of a dictionary and store it in a binary format, it is easier to map the read. So you can easily find where the sequence of the read is matching the genome and map it to that. So if you want to go to the next step, of course you have to use the bwa-mem command which is used to map the reads. So this is a bwa-mem algorithm. Of course there are other mapping options available in BWA which are listed out here, which you may want to use depending on the type of data set that you have, the type of genome that you have, because in some cases if your genome is very small, so for example for a viral genome or something like that, then you may want to use a different algorithm for doing the indexing of the genome. But anyway, let's say you have single end reads. So this is single end reads. So in your single end reads you only have one fastq file. So then what you do is you specify bwa, then you specify the algorithm used for mapping, mem, then you specify the index name or genome name, right. So this command has to be run after the index has been built, and you specify the genome name. After that you specify the fastq file. The fastq file can be gunzipped, right, it can be in a compressed format, and then BWA will uncompress it and use it. So then this output will give you a alignment of these reads to the genome, and it will be given in a text format or in the SAM format. So if you want to compress it, you can just pipe it to the gzip command, and then you can save this compressed version of the SAM file in a gunzip file, right. So this is how you do the read mapping for single end reads. But what exactly is happening? So we already saw that you had your genome, you identified where specific k-mers are occurring, and you built this dictionary which has list of all these positions. So now once you have a read, in the read if the same k-mer is occurring, you see okay, this particular k-mer is present here in the dictionary and it is occurring at these positions. So one of these positions is where this read should get mapped. So instead of looking through the whole genome and deciding where your read should map, you can just look up the positions associated with this k-mer, and you see among these positions what is the best position where your read can be mapped. So by doing this kind of a indexing earlier, you are speeding up the process of read mapping, right. So this is the command for your single end reads, but you may also have paired end reads. So let's say you have paired end reads, then so this for your single end reads. So this for your paired end read, you specify the same thing, you say bwa, then you say mem, then you give the name of the reference genome, then you give the first read, that is the first of the pairs, then you give the second of the pairs. So the two fastq files are given one after the other. Then again of course you can pipe the output to a compression step, and then you can save the compressed alignment in a SAM file which is compressed. But of course when you're doing read mapping, there is one thing that may be useful to add, that is your read groups. Apart from read group, there are various other options you may want to use where you increase the sensitivity of mapping or decrease the number of mismatches that are allowed and things like that, right. So there are various such options that you may want to tweak based on your purpose. But read groups are something which are quite useful to include when you do your read mapping. So what do we mean by read group? So read group is a field which is there in your alignment format, whether it is your SAM or BAM format. So you have this option of read group. So you can think of read group as a kind of a tag which you use to store various metadata associated with the read. So when you map your read, you will get a BAM file, but you may map fastq files from multiple sources and create a single BAM file. So you could have fastq1 from sample 1, you could have fastq2 from sample 2, and all of this could be in one single BAM, right? So you could put all of them in one single BAM. So, but you still want to retain the information about where these reads came from. So some reads came from sample 1, some reads came from sample 2, some reads came from library 1 of sample 1, some reads came from library 2 of sample 1. So all of this metadata that is there, you want to retain it even after you have combined all of it into a BAM file. So this read group is a way of tagging your reads and storing this metadata. So you can store various types of metadata. You can give a unique read group identifier. So this is generally a mandatory field. You need to specify this. Also you specify the sample name. This is again a mandatory field which tells you from which sample is this read actually obtained from. You can also specify library, because within a sample you can have multiple libraries. Although this is not mandatory, you can use this to add extra metadata. You can also provide some description, right? You might want to provide some description which is useful even for somebody who is not familiar with your sample ID or read group ID. Just by looking at the description, they should be able to figure out what this sample refers to. You also can provide the platform, right? Whether you sequenced it with Illumina, you sequenced it with Ion Torrent, you sequenced it with PacBio. So whatever the platform that was used, that can be included. So similar to that, you can also provide the insert size. You can also provide various other details like what was the sequencing center where this read was produced, or the date when the data was generated, right? So all this additional information can be included, and all of this is linked to an individual read. So within the same file, you can have few thousand reads, and each of the read could have different metadata associated with it. So this metadata can be added during the mapping step. So how do you do it? So this is the example. Of course, you use the same BWMM, but you use this -r flag. So -r stands for read group, and when you add the read group, you start with this add symbol, then you say RG, that is the read group, then you give a tab, and then you list out the different type of tags that you can add. For example, you can say ID, and the ID should be sequence 1. Right. So then the ID field is having the value sequence 1 for this particular read. Then you can say sample, and you can say sample 1, then platform is Illumina, then insert size is 300. So all the reads which are there in these two fastq files, when they are converted to SAM, they will have this metadata associated with these reads. Right? All of these reads. But if you want to change the metadata, so of course you can map those reads in a separate command, then you can get multiple SAM, and all of them can be combined into a single BAM, but the metadata is not lost. Right? So once the metadata is linked to a read, it's always going to be linked to those reads. So this kind of a metadata addition in the form of read groups can be quite useful when you generate your read alignments. We will see in one of the subsequent lectures how this kind of read group information can be quite useful when you are analyzing the data subsequently. Right. So this gives you some of the basic operations that you can do when you download data from the short read archive and try to make sense of it. But what are the different uses of these data sets that are submitted to SRA? Because data is being submitted by people from all over the globe, right? People are generating data for whatever different experiment they are trying to do, but then this availability of data which is freely available, which is open to anybody, this allows independent replication. So if somebody has published a study from some other place, right, and they are claiming, okay, this is what we have discovered, and this is the data that is supporting this discovery. So you can download that data, you can try to replicate the results and see if they are really matching your publication, right? So this is one of the things that is facilitated by this short read archive. Also, if you have data sets from different places, right? Like the same kind of a organism is, let's say, sequenced from 10 different countries, and you want to take all these multiple data sets and you want to see how related they are or how they all fit together, right? So then you can take these data sets from multiple sources and you can perform some sort of meta-analysis, right? So for example, you have some patients who are suffering from some specific type of cancer in 10 different countries. You can take all these data sets and see if this type of cancer is having a very similar signature in all these 10 different countries, or are there some differences which are country specific, right? So this kind of a follow-up can be done since all this data is available on one particular platform. Of course, when you have data on a short read archive, it doesn't mean that it has to be only from Illumina, or it has to be only from BGI-seq or only from IonTorrent. There can be different technologies that can be used to generate the data. So the same experiment could have been done using Illumina, the same experiment could have been done using PacBio. And of course, as we saw earlier, there are some challenges or limitations that are there for every technology. So if the same experiment is done with 3-4 different technologies, by comparing the results or comparing how different the outputs are when you use these different technologies, you can try to distinguish what is the real biology and what is the technical artifact associated with some platform, right? So this is again another useful thing, that SRA allows hosting of different type of technology platforms. Then of course, when your data sets which are deposited in SRA, they are generated with some limited set of goals, right? So when they are trying to do that project, they have a very specific focus and they are generating that data to do that very specific task, right? But then later on, when you look at it, you may realize, okay, you can use the same data to answer very different questions. So by reusing this kind of data, you could actually answer some very interesting questions which probably was not possible at the time when the data was generated, right? So this again makes this data much more useful and something which has long-term utility, right? And since this data is stored on SRA, right, it's somewhat decentralized in the sense that data can be generated by very different people, all of it is stored on SRA, and since it is stored on SRA, the data generators are not monopolizing the analysis. So you can actually have any other person analyze this data to share it in whatever way and actually promote various downstream analysis which this data sharing will allow, right? So of course, this has been going on for quite a few years now, and as more and more data sets are being generated, they have been deposited on SRA, and this has stimulated a lot of such diverse discovery and illuminated new biology. So we'll stop with this lecture here. We'll continue with visualizing some of these data in the next lecture.